Picture a busy metropolitan city with people chatting on phones, commuting by train, working in offices. This productivity relies on smooth communication networks functioning properly. Now imagine catastrophe strikes. Cell towers fail, trains halt, offices descend into chaos. Messages can't get through. Our trillion neurons face a similar crisis when their communication network breaks down, leading to a cellular catastrophe that disrupts brain function. Today, we'll explore how a phenomenon called RNA dysregulation can sabotage neurons much like a metropolitan meltdown. By understanding the science behind it, we'll gain clues to overcoming communication disruptions underlying brain diseases like Alzheimer's and ALS. Join me as we dive into the intricate molecular landscape within our minds. Let's unravel this cellular puzzle. It's time to learn what goes wrong in RNA dysregulation. What is RNA? To understand RNA dysregulation, we first need to know, what does RNA do in our cells? RNA acts as a messenger inside our neurons. It carries genetic instructions from DNA and delivers them to structures called ribosomes. The ribosomes then read these RNA messages to construct proteins. Proteins are the essential functional molecules that allow neurons to thrive. You can think of RNA as containing postal letters with instructions for building different proteins. Without proper delivery of these RNA letters, neurons won't get the right packages. RNA binding proteins help regulate this whole postal system. They identify, sort, and deliver the RNA messages to the ribosomes. It's like the RNA binding proteins are postal workers ensuring the letters reach the right addresses. They keep the neuronal postal system running smoothly. But sometimes there are disruptions in this well-oiled machine. The RNA binding proteins start to make mistakes regulating RNA traffic flow. This breakdown in RNA regulation is known as RNA dysregulation, and it can have disastrous effects on neuronal function, as we'll see next. RNA granules. RNA binding proteins often work together in clusters called RNA granules to coordinate all the RNA traffic. Think of busy postal workers sorting masses of mail in a post office. The granules assemble temporarily to organize the mRNA messages. One type of granule that's very important for neurons are stress granules. These form as needed during cell stress to deal with excess RNA. Stress granules are like temporary holiday postal hubs. They help manage the overload but then disband when no longer required. Normally, RNA granules are highly dynamic, rapidly assembling, functioning, and dispersing as needed. But in neurodegenerative diseases, they become dysfunctional. Mutations can make RNA-binding proteins misfold and clump together. Imagine sticky postal workers jammed around sorting machines. These clumps severely congest the RNA granules, preventing proper processing. It's like a 30-car pileup blocking the post office. With key RNA messages blocked, neurons can't adapt to changes. Without smooth RNA regulation, the cells start to degenerate, causes of dysfunction. Earlier, we described how RNA-binding proteins can misfold and congest granules, but what causes this clumping? One factor is mutations in the genes coding for the proteins. These errors can make them fold abnormally like origami swans with bent necks. Cellular stress also overloads the granule system. Think holiday packages flooding postal hubs. Stress promotes protein aggregation. Problems with the cleanup crew allow buildup too. Cells have waste trucks that normally clear granules. But glitches in this disposal process let clusters accumulate. The result is extensive RNA granule congestion and stalled traffic flow in neurons. Just like road blockages in a city, this gridlock can lead to deterioration. Understanding the underlying causes gives scientists clues to potential solutions, as we'll discuss next. Seeking solutions. The good news is researchers are actively exploring ways to prevent or reduce harmful RNA dysregulation. They are developing drugs to boost cellular waste clearance and stabilize proteins, keeping them from clumping. Gene therapies may someday edit genetic errors so proteins fold normally, and engineered RNAs could tag dysfunctional granules for removal. Understanding the underlying biology provides clues to address the root causes. Science is on the case investigating innovative solutions. In the meantime, we can support our natural defenses against RNA dysregulation through lifestyle choices. Regular exercise improves stress resistance and waste removal in our cells. Stress reduction activities like yoga avoid overtaxing systems. Good sleep allows neuronal maintenance and cleanup. Nutritious foods provide vital building blocks for optimal function. Omega-3s protect neurons, while antioxidants combat free radicals that contribute to cellular damage. That concludes our exploration of RNA dysregulation and the communication breakdowns it can cause in neurons. I hope this video provided an accessible understanding of these intricate molecular processes that impact our brain health. If you found this topic interesting, please let me know by giving this video a like and subscribing to the channel. Your support means so much and will help us continue producing more content in the future. For now, thank you so much for watching. Your time is greatly appreciated. Please take care and remember, knowledge is power.